4.5.2 Effect of insect herbivores on plant distribution, abundance and community composition. There is convincing evidence that insect herbivora reduces the productivity, performance and reproduction of a great variety of plant species, including agricultural crops Marquis 1992, Crawley 1997, Carson and Root 2000, Kranz 2005, Ork 2006. Moreover, herbivora can directly compromise a plant's competitive ability by reducing leaf and root growth, Luda et al. 1990, Carson and Root 2000, Hamba diaresis CK and Beckerman 2003, Agrawal 2004. We saw in our earlier discussion of plant defense that herbivory also selects for investments in constitutive secondary metabolites, and thus influences a plant's competitive profile in evolutionary time via inherent constraints and growth. So, it should not be surprising that insect herbivores can play a powerful role in determining the abundance and distribution of plants, as well as influencing the composition of the plant community. An historic example of the biological control of an invasive weed demonstrates what a powerful force insect herbivora can be in affecting plant distribution, Huffaker and Kennett 1959. In the early 1900s, Klamath weed, Hypericum perforatum hyperacaceae, was introduced from Europe into Northern California and Oregon, and by 1945 it rendered over 4 million acres of rangeland unfit for grazing livestock. In the late 1940s, the leaf beetle Chrysalina quadrigemina, Chrysomelody, was imported from Europe and released on infested rangelands. This beetle soon became established, and by 1956 it had largely eliminated Klamath weed in the western United States. Today, small pockets of the weed remain in shady sites where beetle development and survival are reduced, and there is little dynamic evidence that this beetle remains largely responsible for the weed's restricted distribution. Insect herbivores can also have profound effects on the abundance of their host plant and the plant community at large in natural systems, Carson and Root 2000. Specialist leaf beetles such as Microhopola vittata, Chrysomelody, often erupt and defoliate the goldenrod, Solidago altissima, Astraceae, in old field habitats in New England. Damage inflicted by this outbreaking beetle drastically reduces the biomass, survivorship and reproduction of S. altissima. Moreover, herbivore exclusion promotes the formation of dense stands of goldenrods, under which the abundance and diversity of other plant species are significantly reduced. Overall, M. vittata functions as a keystone species because beetle herbivora indirectly increases the abundance of invading trees in goldenrod-aplodid habitats, thereby increasing the rate of succession by speeding the transition of old fields to tree-dominated communities. Another example of how insect herbivora influences plant succession comes from studies on Mount St. Helens in Washington state following the eruption of this volcano in 1980, Fagan and Bishop 2000, Fagan et al. 2005. The eruption created a large area of primary successional habitat on the north slope and extirpated all plant and animal species from the area. Shortly thereafter, the nitrogen-fixing Lupinolupinus lepidus fabaceae colonized the barren area from remnant populations elsewhere on the volcano and subsequently spread rapidly from its initial invasion focus. By 1990, the lupine population consisted of a large, central core region of extremely high lupine density surrounded by numerous, low-density edge patches. However, by 1992, lupine growth rates in recently founded edge patches dropped to levels far below the extraordinary rates observed during the early stages of colonization, contributing to surprisingly slow rates of spread in recent years. Manipulative experiments suggest that insect herbivores were responsible for the slow rate of lupina spread. For instance, the removal of the dominant herbivore Philotyma Lepidoptera Guleckiidae increased the growth of existing plants and the production of new plants in the edge region, thereby accelerating the lupine's rate of increase at the front of the lupine reinvasion. In this system, herbivore gene decreases in lupine reinvasion will likely result in reductions in rates of soil formation, nitrogen input and entrapment of seeds and detritus that are likely to alter the trajectory of primary succession. Seed and seedling feeding insects are also thought to influence the distribution and diversity of plants by affecting where seedling recruits are most likely to survive. Decades ago, Janssen 1970 and Connell 1971 proposed that herbivores and pathogens were key factors in maintaining the high tree species diversity characteristic of tropical forests. They both hypothesized a disproportionately high mortality of seeds and seedlings near mature trees because host-specific seed predators, herbivores and pathogens are more likely to concentrate their attack where the initial seed input is highest. Thus, due to density-dependent or distance-dependent responses of herbivores, the survival of conspecific seedlings was expected to increase with distance from the parent tree, a prediction now referred to as the Janssen-Connell hypothesis or the escape hypothesis, Howe and Smallwood 1982, Clark and Clark 1984, Givnish 1999. Moreover, with high seedling mortality near the parent tree, the probability that other plant species would establish in these sites increases, thus enhancing tree species diversity. Janssen 1970 envisioned seed input and the probability that a seed or seedling will mature, escape a herbivore or pathogen, as functions of distance from the parent tree, figure 4.17. Moving away from the parent adult, seed density should decrease because of dispersal limitations, and the probability that a seed or seedling will be missed by a herbivore, seed predator, should increase. The interaction between seed density and seed mortality predicts maximum recruitment at intermediate distances from the nearest conspecific adult. Candidate mortality agents in the neotropics that are both abundant and known to inflict density-dependent mortality on the seeds and seedlings include seed weevils Coleoptera, Bruchidae, plant pathogens and vertebrates, Janssen 1975, Augsburger 1984, Howe 1986. A study of the neotropical tree Dipteryx panamensis fabaceae supports the expectations of the escape hypothesis in that seedling survival was positively correlated with distance from the parent tree and negatively associated with seedling density, Clark and Clark 1984. Likewise, pod infestation of the neotropical tree Cassia biflora fabaceae by the bruchid beetle Acanthocylides obrionorum, Coleoptera. Bruchidae was strongly density-dependent. Likewise, pod infestation of the neotropical tree Cassia biflora fabaceae by the bruchid beetle Acanthocylides obrionorum, Coleoptera. Bruchidae was strongly density-dependent. Cylinder 1978. Many other studies, but not all, find support for the escape hypothesis. Clark and Clark 1984, Wills et al. 1997, Nathan et al. 2000, Norgauer et al. 2006. However, there has been mixed support for the specific expectation that seedling recruitment is highest at an intermediate distance from the parent tree. Condit et al. 1992. Although heavily critiqued over the last few decades, the Janssen-Connell hypothesis has gained recent support with new evidence for the density-dependent mortality of seeds or seedlings and its effect in maintaining plant species diversity. Wills et al. 1997, Gibnish 1999, Nathan et al. 2000. However, seed predation does not increase toward the tropics and many temperate tree species also experience density-dependent seedling mortality. Hill Riss Lambers et al. 2002, Moles and Westaby 2003. Thus, unless the strength of density-dependent mortality varies with latitude, this factor alone is not likely to explain the high diversity of tropical forests. Besides escape from density or density-dependent seed and seedling mortality, escape hypothesis, seed dispersal can be advantageous by promoting the colonization of suitable, but unpredictable, germination sites such as forest gaps or disturbed roadsides, colonization hypothesis, or by directed dispersal to particular sites with a relatively high probability of survival, directed dispersal hypothesis, Howe and Smallwood 1982, Wenny 2001. Support for the colonization hypothesis comes from weedy plants that broadly disperse many small seeds to colonize disturbed habitats where IP distance competition is initially low. Birds, mammals and ants that cache or move seeds and nuts in specific sites provide support for the directed dispersal hypothesis. Needless to say, these hypotheses are not mutually exclusive because all three usually predict more successful recruitment away from the parent plant. Teasing apart the factors contributing to spatial patterns of seedling recruitment and survival is an area ripe for future research endeavors. Nonetheless, given that seed and seedling survival increase with distance from the reproducing parent, it is not surprising that plants have evolved a diverse array of seed and fruit adaptations that ensure dispersal by wind, water, explosive seed pods and animals, including insects. Howe and Smallwood 1982, Howe 1986, Wenny 2001. Many cases of seed dispersal involve mutualisms between plants and animals, in which animals disperse seeds or fruits in exchange for a reward. Howe and Smallwood 1982, Van der Pyle 1982, Howe 1986, Rogerson 1998, Boyd 2001. Mutualistic seed dispersal often involves vertebrate animals such as mammals and birds, and less commonly insects. Of the insects, ants are by far the most important group of seed dispersers. Stiles 1992. Most cases of ant-mediated seed dispersal involve the collection of seeds or fruits that have aliosomes attached to them, lipid-rich structures that offer a nutritional reward. Rogerson 1998, Boyd 2001. Ants collect aliosome-bearing seeds and carry them to their nests where they consume the aliosome and then leave the seed underground or discard it from the nest. Ant-dispersed seeds are less likely to suffer vertebrate predation because they have been moved to low-risk sites and because some ant-dispersed seeds are tough and less vulnerable to attack from other insect herbivores. On the whole, insect herbivores clearly affect the distribution and abundance of plants. We have seen that the density-dependent attack of seeds by insect herbivores directly influences where seedling recruitment occurs, and insects also contribute to the multitude of seed mortality factors favoring seed adaptations that encourage dispersal away from the parent plant. Ants are often agents of dispersal in some cases and actually move seeds to sites where germination and survival are higher. We have also seen that insect folivores can have striking effects on plant survival, succession, distribution and the structure of the plant community once seedlings have established. In an even more general sense, there is little question that insect herbivores and plants influence each other's abundance, distribution, evolution and diversification. 4.6 The evolutionary ecology of plant-insect interactions. There is irrefutable evidence that plants and herbivorous insects have influenced each other's evolution. Mitter et al. 1988, 1991, Futuma and Keyes 1992, Herrera and Pelmer 2002, Thompson 2005, Winkler and Mitter 2007. The selective effects of herbivores on the evolution of plant defenses on the counteradaptations of herbivores to the vast array of phytochemical and morphological defenses stand in testament to reciprocal evolutionary effects. Moreover, plant defense theory is premised on the assumptions that plant defenses reduce herbivora and that herbivora is an important selective force underlying the evolution of secondary metabolites. What is far less clear is the extent to which plants and insect herbivora have directed each other's diversification and what conditions influence the degree of coadaptation and coevolution. Implicit in models of coadaptation and coevolution is that herbivorous insects not only promote plant genetic diversity, but they ultimately shift and adapt to novel plant genotypes with consequences for their own diversification. Farrell and Mitter 1990, Mitter et al. 1991, Herrera and Pelmer 2002, Thompson 2005. Thus, factors influencing host plant specialization, shifts to novel plant genotypes or species and diet breadth evolution, monophagy versus polyphagy, are central to theories on lineage diversification, coadaptation and coevolution. Plant-insect associations provide an ideal model for examining the process of diversification because phytophagy promotes higher rates of diversification than other feeding stylistic as predation and saprophagy. Mitter et al. 1988, Wiegmann et al. 1993, Schluter 2000, Winkler and Mitter 2007. This assertion has been rigorously supported using replicated phylogenetic contrasts between sister lineages, which by definition are of equal age, an assessment that found Phytophagus lineagisto far more species-rich than Thernan Phytophagus counterparts. We will explore these issues in greater depth in the sections that follow. 4.6.1 Host shifting and diet breadth evolution. Earlier in this chapter, we learned that most Phytophagus insects, greater than 70% of species, are specialized in their use of host plants, monophagous. Historically, dietary specialization and host shifting have been thought to promote herbivore diversification, Futuma and Keyes 1992. Two factors contribute to this expectation. First, a given plant diversity represents a far greater variety of resources for specialist herbivores than generalists, thus providing greater opportunity for reproductive isolation and speciation. Second, specialization might promote herbivore diversity if the property of specialization enhance the likelihood for speciation. For example, an accidental colonization of a novel host plant might foster specialization and speciation if mating occurs exclusively on the host plant, Janik 1990, Kaloud and Via 2000, Berlacher and Feder 2002. Alternatively, it has been argued that polyphagy facilitates radical host shifts because less specialized species are more likely to make oviposition mistakes on novel hosts, thus enhancing opportunities for diversification, Jans et al. 2006. Because diet breadth is so labile with reversals between monophagy and polyphagy in the SAMA lineage of herbivores, diet breadth is not likely subject to much phylogenetic constraint, Nosel and Moores 2005, Yotoko et al. 2005, Winkler and Mitter 2007. Thus, standard phylogenetic techniques may be less able to shed light on diet breadth evolution, such as whether specialization evolves from a polyphagous lifestyle following colonization of a novel host. Counter to the expectation that specialization fosters diversification is the finding that, in a significant majority of 20 phylogenetically controlled comparisons, lineages of concealed feeders, e.g., borers, were less diverse than their external feeding sister clades, despite the fact that internal feeders tend toward greater host specificity, Winkler and Mitter 2007. Regardless of the underlying mechanisms and the ancestral state of diet breadth, a recent phylogenetic assessment suggests that shifting to novel hosts per se plays a major role in promoting the speciation of Phytophagus insects, Winkler and Mitter 2007. Given their importance in the diversification of Phytophagus insects and in theories of coadaptation, let's now examine the various factors that are rethought to shape the diet breadth and host shifting in Phytophagus insects. The relationship between the oviposition preference of females for particular host plants and the performance of their offspring on those hosts has played a central role in the evolution of diet breadth in herbivorous insects, Futuma and Moreno 1988, Thompson 1988, Janik 1990. However, other factors, such as host plant abundance, risk of enemy attack and mating on the host plant have been suggested as important contributors to diet breadth as well. In the paragraphs that follow, we will explore the many factors that have been proposed to influence diet breadth in insect herbivores, Janik 1990, examine the evidence for each and then highlight which factors appear to be most influential in promoting specialized or broad diets. See also Chapter 8 on host and parasite interactions. 4.6.2 Selective factors favoring narrow herbivore diets. Because of allochemical and other dissimilarities among plants, most insect herbivores are not likely able to use the vast array of locally available host-plant species, Janik 1990. For example, monarch butterflies, Danaus plexippus, that rely on unique chemical cues to locate their milkweed hosts, and whose larvae are adapted to cope with the toxic cardenolides therein, are not likely able to either find or develop unintroduced eucalyptus trees with vastly different allochemical signatures. Thus, at a gross plant taxonomic scale, one expects to find a positive relationship between oviposition preference and offspring performance. It was observations like these that led ecologists to view a positive preference-performance correlation, namely females preferring to oviposit on plants where their offspring grow and survive best, as a potentially powerful factor favoring dietary specialization, Thompson 1988, Janik 1990. However, with more restricted sets of host plants, such as congeners or plants in closely related genera, there has been mixed support for a positive preference-performance relationship, Janik 1990, Abramson and Weiss 1997, Mayhew 1997, Agrawal 2000b. Indeed, some herbivores rank host plant species for oviposition in the same order as their suitability for offspring performance. Examples include the heliconiid butterfly Agralis vanilli on passiflora species, the Cytomyiid fly Rhabdophaga terminalis on willows, and the agromyzid leaf miner Liriomyza sativate on agricultural crops, Kopp and Davenport 1978, A. H. Mann 1984, Via 1986. However, many other insect herbivores do not show this positive association, Thompson 1988, Abramson and Weiss 1997. Low correspondence between preference and performance can arise when females fail to oviposit on plant species that are perfectly suitable for larval development, as occurs in the swallowtail butterfly, Papilio machaeon, Wickland 1975. In such cases, selection may have favored larvae that accept a wider array of host plants, while at the same time adults oviposit only on the most suitable hosts in a local patch. Alternatively, females may fail to oviposit on novel introduced plants that are suitable for larval development, simply because they fail to recognize them in the absence of appropriate oviposition cues. Poor correspondence between preference and performance can also arise when females oviposit on host plants where larvae fail to survive or develop very slowly. Such a case occurs in Rocky Mountain populations of the mustard white butterfly, Pieris napi, which oviposits on 7 crucifer species including 2 introduced species, Thalaspi arvens and Chorosporotinella, with glucosinolate profiles similar to those in native hosts, but that are lethal to larvae because of additional toxic constituents, Chu 1977, Rodman and Chu 1980. Likewise, females may oviposit on poor quality host plants because their offspring obtain protection from natural enemies. For example, the softline Neodyprian certifer prefers to oviposit on pine trees with high resin acid content, Biodiaresis R.K. Mann et al. 1997. When natural enemies are excluded, larvae perform poorly on resin-rich trees, whereas in open cages larvae suffer far less mortality from enemies than they do on resin-poor trees because the regurgitated resin is used as an effective defense against parasitoids. Thus, when risk of enemy attack is diminished on plants where performance is reduced, there may appear to be poor correspondence between oviposition preference and performance when performance is assessed in the absence of natural enemies. It is important to note that there is far better correspondence between preference and performance when herbivores choose among plants of the same species, Janik 1990, Price 2003a. Many aphids, planthopers and sawflies, for example, selectively oviposit on plants where offspring performance is high, Witham 1983, Cook and Dino 1994, Dino et al. 2002, Price 2003a. In several of these cases, increases in soil nitrogen created high-quality patches off plants that were both preferred for oviposition and on which offspring performance was enhanced. Thus, it is important to realize that environmental factors can influence the preference-performance relationships of insect herbivores. Another potentially powerful factor promoting host plant specialization is a physiological trade-off in herbivore performance on different host plant species that is evidenced by a negative cross-host correlation in performance, Janik 1990, Futuma and Keyes 1992, Thompson 1994. Intuitively, if the biochemical machinery necessary to detoxify allochemicals found in one host plant precludes this ability on another, then such a negative correlation should favor monophagy. One of the few earlier studies that examined performance relationships featured the fall canker worm, Alsophila pomateria, Lepidoptera, geometridae, and its larval performance on several co-occurring tree species, Futuma and Philippi 1987, Janik 1990. Larval clones that grew faster on red maple grew slower on several oak species, providing support for a host-related trade-off in larval performance. More recently, the host races of the planthopper Niliparvata lugans on the native grass Lyrcia hexandra and on rice, Oryza sativa, were reinforced by a negative trade-off in performance on the two host plants, Cesar and Butlin 1998. However, other studies seeking support for a negative correlation in cross-host performance have found either no relationship or a positive correlation, such that high performance on one host promotes the same on another, Bernays and Graham 1988, Janik 1990, Fry 1996, Thompson 1996. Earlier studies on preference-performance relationships or cross-host performance correlations were largely phenotypic and were hampered by a nearly complete lack of information on how these processes were genetically related, via 1986, Thompson 1988. Moreover, it is undeniable that an understanding of the genetic underpinnings of preference and performance traits is central to elucidating factors influencing diet breadth evolution. Several recent studies on aphids find convincing support for genetically based correlations in preference and performance that strongly favor host plant specialization, Hawthorne and Via 2001, Via and Hawthorne 2002. For instance, highly specialized host races of the pea aphid, Acyrthosyphon pisum, occur on alfalfa, Medicago sativa, and red clover Trifolium praedens. Using a quantitative trait locus (QTL) analysis, evidence was found for both a negative correlation in aphid performance fecundity on the two host plant species and for a positive association between host plant acceptance and performance. A positive genetic correlation between preference and performance could arise by pleiotropy, the same allele affecting both preference and performance, or linkage disequilibrium, alleles affecting preference and performance occurring in close physical proximity on chromosomes, thus increasing their chances of being inherited together, Falconer and McKay 1996. In the above aphid example, genetic correlations could be generated by QTL that have either pleiotropic effects or linkage disequilibrium. Not all genetic assessments, however, have found convincing evidence for either a positive association between preference and performance or for a negative correlation in performance across host plants, Joshi and Thompson 1995, 1997, Fry 1996, 2003, Thompson 1996, Abramson and Weiss 1997, Balabeni et al. 2003. Natural enemies can also shape the diets of insect herbivores and promote specialization. For many insect herbivores, susceptibility to predators and parasitoids varies as a function of the host plant species they consume, Bernays and Graham 1988, Janik 1990, Burdig et al. 1996. If, for example, herbivores sequester specific plant toxins and use them for defense against generalist predators, the enemy free space EFS provided could promote dietary specialization by restricting herbivores to plants where toxins can be obtained. Jeffries and Lawton 1984 define enemy free space as ways of living that reduce or eliminate a species' vulnerability to one or more species of natural enemies. In fact, seldom is an area of a species niche totally free of natural enemies, and studies generally reveal the existence of enemy-reduced space, Burdig al. 1996, a more accurate moniker for this widely used concept. An example consistent with the hypothesis that EFS promotes dietary specialization in bulbous leaf beetles Photora vitellinae and Galericella lineola that feed on willows in Scandinavia, Dino et al. 1990. When disturbed by a predator, larvae of P. vitellinae exude droplets of a defensive secretion from dorsal glands. The secretion contains salicylaldehyde, the precursors for which are plant-derived salicylates like salicin phenolics. When fed willow species rich in salicylates, larvae defend themselves effectively against predators such as coccinellid beetles. In contrast, when fed salicylate-poor willows, larvae are readily consumed. Herbivoresic as P. vitellinae obtain enemy-free space in willows where they can acquire the allochemical precursors for defense. The result appears to be dietary specialization on salicylate-rich willow species, even though they can survive and develop well on salicylate-poor species in the absence of predators. Related herbivores like G. lineola that do not employ a plant-derived defense are not so constrained and use a broader range of willow species in the field. Other evidence that predation can shape the diet breadth of herbivores comes from an experiment involving a taxonomically diverse array of 70 species of neotropical caterpillars offered to the predaceous ant Paraponera clavata, Dyer 1995. Overall, specialist caterpillar species that either sequestered plant chemicals or synthesized them de novo were far more protected from ant predation than palatable generalists. Moreover, when specialists that normally sequester plant toxins were reared on toxin-free diets, they became more susceptible to attack than larvae reared on leaves with access to toxins. Thus, both plant chemistry and predators may act in concert to select for dietary specialization in this assemblage of herbivores. As a caveat, however, chemically defended caterpillars that are protected from predation in this assemblage are susceptible to attack from parasitoids, Gentry and Dyer 2002. In fact, specialist lepidopterans at large incur higher attack rates than do more prolificous species, Dyer and Gentry 1999. Thus, the advantage specialists enjoy from relaxed predation may be offset by increased parasitoid attack. It should be noted also that there are highly prolificous species, such as the lubber grasshopper, Romalea microtera, that sequester toxins from a wide variety of plants and lower their risk of predation by so doing, Bloom et al. 1990. Thus, EFS achieved by toxin sequestration may not always be associated with dietary specialization. Crypsis camouflage by resemblance to the background environment can also promote narrow diets as a consequence of enemy-free space, Ott and Jorn 1977, Janik 1990, Crespi and Sandoval 2000. In the case of desert grasshoppers, Orthoptera, acrididae, species that resemble the specific background of their host plant gain protection from visually orienting predators, Ott and Jorn 1977. As a consequence, adaptation to one host background diminishes protection on another and the specificity of crypsis is inversely related to diet breadth. For example, Budetix grasshoppers that live exclusively on and precisely resemble the foliage of their Laria host plant, Zygophyllaceae, are very host-specific. In contrast, crypsis in stem-inhabiting grasshoppers, e.g. Ligurotetex coquilleti, provides effective camouflage across a variety of habitats and these grasshoppers are far more prolificous. Similarly, Tyma walking sticks Phasmatodea, Tymemity gain protection from vertebrate predators, lizards and birds by resembling their host plant, Crespi and Sandoval 2000. In this assemblage of walking sticks, selection for crypsis has resulted in dietary specialization. In fact, certain species, Tyma cristinae, have evolved different plant-associated color morphs, a polymorphism that is maintained by differential risk of predation of the morphs on the respective host plants. Also, host shifts to distantly related plants have occurred because certain walking stick species possess color patterns that pre-adapt them for crypsis, and therefore enemy-free space, on novel plants. Interspecific competition is another factor that may favor dietary specialization, Janik 1990. By specializing on different plant species, herbivores could reduce competitive interactions that otherwise might result in reduced fitness, Dino et al. 1995. This assertion assumes that competition is a consistent and important source of mortality for herbivores, a view that has attracted considerable debate, but is regaining momentum, see Chapter 5. Nevertheless, the great majority of studies testing for interspecific competition between herbivore species involve interactions on the same host plant species, thus precluding an assessment of how competition might restrict a herbivore to using a subset of available plant species, Dino et al. 1995, Dino and Kaplan 2007, Kaplan and Dino 2007. One study, however, sheds light on the issue. For instance, the survival of the plant bug Megaloceroea recticornis was reduced by another plant bug Nodostera elongata when only a single host plant species was present, but the competitive effect was removed in the presence of an alternate plant species that acted as a refuge for the inferior competitor, Gibson and Visser 1982. This dietary shift is consistent with the view that interspecific competition can be minimized when co-occurring herbivores specialize on or occupy different host plant species. Host plant abundance and apparency can also influence the diet breadth of insect herbivores, Courtney and Forsberg 1988, Singer et al. 1989, Janik 1990, Kuasari et al. 2000. Specializing on host plants that confer high offspring fitness is favored only if the realized fecundity of the female isn't compromised by other factors such as plant rarity. As abundance of an acceptable host plant species increases in the habitat, herbivores evolve oviposition preferences for the common host, which often translates into increased use. A good example involves the glandular fritillary butterfly, Melidea synxia, Lepidoptera, Nymphalidae, that selectively oviposits on Veronica spicata as this host plant becomes more abundant in the habitat relative to an alternative host plant, Lanceolata, Kuasari et al. 2000. Alternatively, when the most suitable host plant species is rare in the habitat, females move down the preference hierarchy and oviposit on less favored hosts, Thompson 1988, Singer et al. 1989. This said, oviposition preference is a complex process and is influenced by a variety of herbivore-specific factors, including genetic variation in preference rankings among females, a female's motivational state and egg load, learning and age, Janik 1990, Radke Anzinger 1995, Singer and Lee 2000. For instance, females are more likely to oviposit on less preferred hosts if they are old and carrying a large load of undeposited eggs. Regarding learning, apple maggot fleas are more likely to oviposit on fruit types in which they have had previous experience, Papage and Procopi 1988. Despite the variation imposed by these factors, the spatial use of host plants appears to be driven largely and directly by plant abundance and indirectly by an evolved preference for theoretically abundant acceptable host plant, Kuasari et al. 2000. Plant ephemerality also influences diet breadth. Among Mycophagous drosophilid flies, Drosophila duncani and Mycodrosophila cladoni specialize on a few species of very persistent bracket fungi, whereas D. follini and D. putrida use dozens of species of ephemeral mushrooms. Lacey 1984. Overall, both abundance and persistence of host plants have played an important role in shaping the diet breadth of phytophagous insects. If the fitness of an individual choosing a particular host plant species increases in the presence of other colonizing conspecifics, then host specialization can arise. Janik 1990. An example would be species that choose host plants on which they are more likely to encounter mates. So-called cases of plant-associated assortative mating, mating with individuals like themselves who in this case prefer the same plant, are thought to strongly favor adaptation to specific host plants. Fruit flies in the Ragoletus pomonella complex, Diptera. Tephrididae feed as larvae within the fruits of their hosts and provide a classic example whereby assortative mating has promoted specialization on a variety of trees and shrubs including hawthorn, crataegus, apple, malus, and dogwood, Cornus, Bush 1969, Feder et al. 1994, Procopi and Papage 2000, Berlocker and Feder 2002, Forbes and Feder 2006. Flies in this complex of host races mate solely on fruits of their host plant, and this behavior likely facilitated a shift of R. pomonella from hawthorn to apple in the mid-1800s as apple became more abundant. Host-associated mating minimizes genoflow among the host races, promotes genetically beset differences in oviposition preference, and synchronizes flies' life cycle with the phenology of fruit production, all adaptations that have led to host plant specialization, mating barriers and perhaps speciation. A similar situation occurs with the gall-inducing fly Eurosta solidagenus, Diptera. Tephrididae that forms host races on two species of goldenrods, Solidago altissima and S. gigantea, Astracei, Abramson et al. 2001, Craig et al. 2001, Eubanks et al. 2003. Genetic differences between host races in this fruit fly have resulted from host-associated assortative mating as well as strong oviposition preferences. In this system the predaceous tumbling flower beetle, Mordella stenoconvicta coleoptera, Morellidae that attacks day fly has also formed host races on the two Solidago species. Notably, host-associated mating also occurs in the beetle and has contributed tolical adaptation. This study suggests the interesting possibility that host race formation by a herbivorous insect can promote subsequent diversification at higher trophic levels, a phenomenon referred to as sequential radiation by Abramson et al. 2003. Plant-associated mating also encourages specialized diets in other herbivores. For instance, the pea aphid, Acyrthosiphon pisum, mates on its alfalfa and clover hosts where specialization on each plant species is facilitated because genes controlling host acceptance, assortative mating and performance are genetically linked. Kaloud and Via 2000, Hawthorne and Via 2001, Via and Hawthorne 2002. Not only does this genetic architecture promote host plant specialization, but it can also foster reproductive isolation and speciation. Similarly, assortative mating has played an important role in the specialized diets and host race formation of the Enchinoppa binotata complex of treehoppers, Hemiptera. Membracidae, Wood and Gutman 1982, 1983, Wood et al. 1999. These sap feeders exploit a number of tree and shrub species in eastern North America where there is strong evidence for host race formation. Membracids, like their leafhopper and planthopper relatives, have a unique mate location system that ties them very closely to their host plant and thus promotes assortative mating. Both sexes locate and communicate with each other during courtship using substrate-borne vibrational signals they produce. Such signals can be detected only by individuals contacting the same plant through which the signals are conducted. Thus, both mating and oviposition into host plant tissues are closely linked to the same individual plant. Such close linkage has promoted preferences for an adaptation to the specific phenologies of the different host plant species. Assortative mating in Ragoletus flies, pea aphids and Enchinoppa treehoppers has been championed as a prerequisite to host race formation and speciation at a small geographic scale, namely sympatric speciation, Wood et al. 1999, Hawthorne and Via 2001, Berlocker and Feder 2002. Although the process of sympatric speciation has generated considerable debate, it can hardly be contested that host-associated mating can be an important factor promoting genetic divergence and dietary specialization in insect herbivores given that so many often mate on their host plants. Aggregation behavior in herbivores may also promote dietary specialization, Janik 1990. For instance, many species of insect herbivores including aphids, lepidopterans, beetles and sawflies feed in coordinated groups, Bowers 1993, Fitzgerald 1993, Dixon 1998, Fordyce 2005, 2006. Also, many herbivores that feed in aggregations are aposematic, warningly colored, sequester toxins from their host plant and are distasteful to predators. Advantages of feeding in aggregations include increased foraging efficiency, feeding facilitation, defense against predators, thermoregulation and overwhelming plant defenses. To a certain point, all of these factors are enhanced with increasing group size and thus are density dependent. Many, but not all, aggregative herbivores on toxic plants are dietary specialists. For example, several tree-feeding Lepidoptera such as Mollicosoma, Hyphantria, Halizodota and Samurista are gregarious feeders, but are highly prolificous. Baker 1972b, price 2003a pages 155 to 158. An example of how larval aggregation may promote specialization involves the crimson patch butterfly, Clocingenis Lepidoptera, Nymphalidae that feeds only on the shrub Odontoma calistaceum, Acanthocei, in the subtropics of Mexico, Dino and Benry 1997. When experimentally manipulated, there is a striking effect of group size on larval growth whereby larvae more than double their weight gain by feeding in large rather than small aggregations on intact plants in the field. However, this group feeding advantage is lost altogether when larvae are raised on excised leaves in the lab, suggesting that large aggregations may overwhelm an induced allochemical response. Given the intimacy of the interaction required for herbivores to manipulate plant defenses, Carbon and Baldwin 1997, it is unlikely that the benefits of aggregation would be as effective across a broad range of plant species. Thus, aggregation and associated traits such as warning coloration and increased defense against predators may contribute to dietary specialization.